So Dr. Paul Grossfeld is going to present now about his cardiology research. Right after that, we are going to meet outside for the group photo. And the kids and the care team has then prepared a surprise activity for all of us. So that's going to be outside. Thanks.
I'm going to walk around a little bit. So. Is this on? It doesn't sound like it's on. On now. Yes. Okay. You going to help me? All right. Well, thank you again for inviting me and Susan to be here. It's really an honor and a privilege. And I have some pretty exciting developments to update you on, even since our last conference. A couple of things. First of all, some of these slides have a lot of technical details. Don't worry about that. I'm just going to try to provide some summaries about some of those slides. Also, I do have a few pictures of actual human hearts, and if anyone is uncomfortable about that ahead of time, I'll kind of warn you so you can close your eyes or look away, but they're actually pretty cool looking. So, and lastly, again, feel free to stop me at any time to ask questions. We're all among friends here. This is not like a lecture per se. So, all right. So what I wanted to do is actually start out by sharing kind of my personal experience back when I was in medical school and graduate school, and to tell you the story about how I met these two physician scientists and what an influence they had on me that I've been able to kind of use their example to implement the approach that I've taken to studying congenital heart defects and Jacobson syndrome. So on the left is from 30 years ago, Mike Brown and Joe Goldstein. This is where I was in medical school in Dallas. And back when they were in their training, they were very interested in trying to understand how about 1 in a million children at age about 5 have a heart attack due to blocked coronary arteries. And that 1 in a million child has a cholesterol level of something like 1,000. Now in the United States, normal cholesterol is maybe 150, 200, so way off the charts. And so they started by studying a very rare syndrome or disease, which actually has gone on to be called familial hypercholesterolemia. And when I was there in Dallas, they actually won the Nobel Prize for their work. And what they ended up accomplishing is they were able to work out the whole pathway for how our body either takes up cholesterol from the diet and or how cells actually produce cholesterol and how that balance between diet and synthesis is balanced, how that balance is achieved. And this little girl was kind of the first beneficiary of their research because the liver has an important part in managing cholesterol, both taking it up from the diet and synthesizing it. And so she actually was effectively cured in her case by getting a liver transplant. More importantly, though, the pathways that they discovered for how cells make cholesterol led to the development of this drug, which up until recently has become the most commonly prescribed drug in the world that probably many of you, including myself, are familiar with, called Lipitor. So that was a great model that they started with something very rare, and the hope was what they learned from that very rare situation could apply to a much broader population. All right, so what am I interested in? My interest as a pediatric cardiologist has been to understand how this heart defect happens. And so this is just a schematic diagram showing a normal heart, normal left ventricle chamber, right ventricle. These are the two valves on the left side, the mitral valve and the aortic valve. And this is the aorta, the main vessel that supplies blood out to the body. So that's a normal heart. The defect that I've been most interested in is something that you've heard me refer to called hypoplastic left heart syndrome. And here you see that instead of this nice big chamber that fills with blood, it's very, very small. Excuse me. And the way it's shown here is it looks like there's almost been overgrowth of those walls inward to kind of obliterate that chamber. And going along with that is very small, sometimes completely blocked off mitral valve, aortic valve, and compared to the normal aorta, this aorta is very, very small. And as you can imagine, that is not compatible with life. And I won't go off into how we manage these kids, but there's no cure. And actually, hypoplastic left heart syndrome is the most common cause of death in all infants with any kind of heart defect. And I think now it's the most common cause of death in infants with Jacobson syndrome as well. Let me know, translators, if I'm going too fast. Doing okay? Slow down. All right. I already kind of mentioned this. And just from an economic standpoint, in the United States, there are about maybe 1,000 to 2,000 babies per year with hypoplastic left heart syndrome. And the direct costs, I'm sure, are in excess of $1 billion a year. Not to mention the indirect costs of families losing time from work, the psychological impact of having a child with a life-threatening heart defect takes quite a toll. All right, close your eyes if you don't want to see anything about a human heart. I showed you that cartoon, but this is the real world of hypoplastic left heart syndrome. And what you see here is actually 4 different variants where the common denominator in all these is a very small left ventricular chamber. And I just want to focus on these 2 on the left. And in this case, what I'm outlining here is what was supposed to be the left ventricle. And this kind of goes along with that cartoon where it really suggests there's overgrowth of that heart tissue to kind of obliterate the chamber. And this white stuff is actually like a form of scar tissue. So that's one form, that's one of the most common forms. And this is the other one, and this is actually the other most common form, and this is the form that I've discovered occurs in children with Jacobson syndrome. And in this case, here is what was supposed to be that left ventricular chamber, so that's why they call this a slit-like left ventricle. And it's hard to appreciate here, but the mitral and the aortic valves are what we call atretic, meaning they're completely blocked off. And as I'll show you a little later on, we think that this heart defect occurs very, very early as the heart is developing, which actually goes against what has been kind of the dogma. But a big problem with the history of research with hypoplastic left heart syndrome is that the few PhDs studying this would all kind of assume these are all the same. They never pay attention to the different anatomic subtypes. But what I am certain about is the mechanism that leads to this overgrowth has to be very different from this mechanism where there's actually lack of growth, underdevelopment. Does that make sense? And I just summarized that here. So the version of hypoplastic left heart syndrome that occurs in Jacobson syndrome is due to something that occurs very early in heart development, probably in most cases even before the mother knows she's even pregnant. So that's a challenge in and of itself. Versus other forms that have been shown in animal models actually probably occur much later in heart development. Let's see. So probably many of you have seen this slide before. This is the little boy that I was taking care of in July of 1995, Edgert. And I was taking care of him in my first month of cardiology training because he had a form of hypoplastic left heart syndrome. And in his paper chart was this picture of his chromosomes. And I think all of you can appreciate that he had a pretty large deletion, missing the end of the long arm of chromosome 11. So in 1995, all I could guess is that there must be something interesting in that region that's deleted in terms of a gene or genes. That must be the cause of his heart problems, and obviously what was determined to be Jacobson syndrome. How to go about it at that point, I had no idea. So, as I've mentioned before, these have been historically the three most common causes of death in people with Jacobson syndrome. I think now we have a much better handle on, at least theoretically, never seeing someone die from bleeding or infection with Jacobson syndrome. And not perfect, but I do think we've made a lot of progress in that over the years. So, as I kind of alluded to the other day, I initially in my training, and since then, even now, have spent as much time as I can learning about all the aspects of Jacobson syndrome. And we published this paper in 2004 with about 110 people. The previously largest study was 17, so this was a big step forward to getting information out there about Jacobson syndrome. And in terms of heart defects, interestingly, not everyone, as you know, with Jacobson syndrome has a heart defect. We found that about half roughly do. And quite to my surprise, compared to any other known genetic syndrome, it has one of the highest frequencies of hypoplastic left heart syndrome, maybe about 200 times the frequency in the general population. So of those 2,000 babies a year born in the United States, maybe 1% to 2% of them have Jacobson syndrome. And as I already mentioned, interestingly, it's most commonly of this one anatomic subtype that I showed you. Another thing that we've learned is that many of the most common congenital heart defects, including what some of your children were born with, also occur in Jacobson syndrome. And another challenge has been to understand why is it you can have two children with the same deletion, one might have hypoplastic left heart syndrome, the other a normal heart. And we've made some progress on understanding that. Anybody have any questions so far? Okay, close your eyes if you don't want to see a human heart. This is actually the heart of a little baby that was born a few years ago in Dallas with hypoplastic left heart syndrome and Jacobson syndrome. The baby passed away on the 13th day of life, and the family graciously donated their baby's heart to my research. And again, I think you can appreciate here that very slit-like left ventricle and why this is not a survivable heart defect. Something that we've also, again, made some progress, which I'll talk a little bit about, is why is it one child has no heart defect, another child has a hole in the heart, and a third child has hypoplastic left heart syndrome. And I'll tell you that the progress we've made, I think, just in the last couple years will help us to answer some of those questions. So probably most of you in this room are familiar with this technology called array comparative genomic hybridization, or CGH, or microarrays. And in the old days, like I showed you with Edgar's chromosomes, that was how it was diagnosed. But that's a very qualitative description. This technology now allows us to actually look at the deletion and get it down, define the breakpoint to the individual gene level. And some of you that I've met with have asked me, well, what does my child's deletion mean? What can I expect? And it's this technology now that allows us to have that level of understanding. So this has been a huge step forward to help us to narrow down the region where these genes are. The other technology that has come out since I started this is the sequencing of the whole human genome, so 3 billion base pairs. And in San Diego, we actually have the technology at our hospital to sequence 3 billion, base pairs and get all the information back in less than a day. The original human genome sequencing project, I think, cost them the billions of dollars for one patient. They're talking about doing this now at a cost of about $100. So that's an incredible achievement in the technology. So using that technology, we were able to take advantage of some select patients that had heart defects and different types of deletions. So this deletion here is the smallest deletion that goes out to the end in a child with hypoplastic left heart syndrome. And compared to Edgar's, whose deletion I think was about 16 megabases, this one narrowed it down to a region of 7 megabases. So already that one patient potentially helped us to narrow a region containing about 150 genes, maybe down to about half that number of genes. And then taking it one step further, we took advantage of some of these even rarer patients with hypoplastic left heart that have so-called interstitial deletions, so within and not at the end. And we hypothesized that if it's the same gene or genes that cause the heart defects in these patients versus this patient, then it must be somewhere in this region of overlap. Make sense? And thanks to the Human Genome Project, we could then narrow that region of overlap to about 6 genes. And what I've put in bold is 2 genes in particular that, at least the first one, already looked like a very obvious candidate. This gene is referred to as a transcription factor, which you've heard a little bit about. So transcription factors turn on and turn off at genes at different times, let's say in development or sometimes in cancer. And actually a second gene, which I'll talk a little bit about, is very similar to this first gene called ETS1. And there were already studies showing that ETS1 has a very important role in the development of the heart of an organism called the C-squirt. So that really was a step forward. That right there summarizes about my first 10 years of work, which these days we could probably get that done in about a month if we were starting out today. Taking it one step further, we also learned about a patient who had kind of a version of hypoplastic left heart syndrome, did not have a deletion, but did have instead just what we refer to as a point mutation in that same ETS1 gene, like a letter in the genetic code like you heard about this morning. And so that provided further evidence implicating the ETS1 gene as at least one gene that can cause these heart defects. And more recently, actually in San Diego, we have a patient who has hypoplastic left heart syndrome. Oops, sorry about that. The same type that typically occurs in Jacobson syndrome. And that patient actually has another point mutation, not in ETS1, but FLY1. And actually, kind of to get ahead of myself, but we actually think it's probably loss of both FLY1 and ETS1 under the right circumstances that will give you hypoplastic left heart in the human situation. So my research lab uses genetically engineered mice to study these genes and their role in normal heart development and what happens when you eliminate the gene in terms of causing a heart defect. Don't worry about all the blue color, but one of the first things we wanted to show is whether ETS1 is actually functioning as the heart is developing in the mouse. And what this picture shows you is in mouse embryos, in the embryonic heart, the blue is showing where the ETS1 gene is being expressed. And what we found is that there are 2 populations of cells in the developing mouse heart that express ETS1. And that's what I'm going to spend most of the rest of my talk telling you about. One of those populations of cells is called the neural crest, and the other population is called actually the endothelium or endocardium, two populations of cells in the heart, and I'll tell you how we've looked at how what their role is in heart development and disease. So basically, what we hypothesize is loss of ETS function in one or both of these populations of cells will cause the heart defect. But what's very interesting though, and this is a really important observation from early on, is that the actual working cells of the heart, those are the cells that do the actual contracting. They're called cardiac myocytes, which is what the ventricle that I showed you before is mostly made up of. ETS1 is not expressed in cardiac myocytes in the developing heart. So there has to be some kind of communication between these other populations of cells and the developing heart muscle, so that if that communication is somehow disrupted, it affects the development of that heart muscle, of the ventricle. So just to review a little bit about the first population, called the cardiac neural crest. The neural crest is actually a population of cells that originate in the developing neural tube or spinal cord of the heart. And what you see here schematically is that as the heart is developing, there is a subset of these cells called the cardiac neural crest that move towards the developing heart to very specific locations. Let's say down here. This is the wall that separates the aorta from the main lung artery. And this is a very highly regulated process that makes sure that these cells know who they are and when they're supposed to go and where they're supposed to go to help form these structures of the heart. So that's the cardiac neural crest. Does that make sense? Nobody's asleep yet, that's good. Now, interestingly, as I mentioned before, my hypothesis is one or both of these populations of cells is kind of the smoking gun. And further evidence is shown here in that there is a type of problem with the intestine, a disease called Hirschsprung's, where the intestine doesn't move properly because of a defect in the neurons. And we know that Hirschsprung disease is due to a problem with neural crest development. And what was published is if you take all children with hypoplastic left heart syndrome, there's a much higher frequency of Hirschsprung's in that population of patients, suggesting that the neural crest cells may have some role, by association at least, in the pathogenesis of hypoplastic left heart syndrome. And so we can actually use our genetically engineered mice to eliminate the ETS1 gene just in either the neural crest cells or that other population of cells, the cardiac endothelium. And I don't know how well you're going to see this, but these are images of an embryonic mouse heart. This is actually using cardiac MRI that some of you have seen or had done as human patients. This little line here is 1 millimeter, just to give you the scale. So this is a normal heart. This is the left ventricle. This gap here is the wall that separates the left ventricle from the right ventricle. And if you have a hole in that, that's called a ventricular septal defect. And you see the aorta is red, lined up over the left ventricle, and the right ventricle connects to the lung arteries. If you look close up at the aortic valve, you see these 3 leaflets. So that's a normal aortic valve. And you also see these blue dots here, or the red dots there. Those are the neural crest cells that have migrated to this developing heart, specifically to actually down here, the septum, as well as the heart, the valve leaflets. When we knock out ETS1 just in the neural crest cells, we see a hole in the heart. We see the aorta that is now lined up with the right ventricle instead of the left ventricle. And in the majority of the cases, we see a very abnormal aortic valve where you only see 2 leaflets instead of the 3 leaflets. And compared to the control, you see much fewer blue cells here and much fewer red cells here, meaning that for whatever reason, there are fewer neural crest cells that made it to those final structures. And this heart defect is specifically called double outlet right ventricle. And one part of that is the hole in the heart, as you can see here, interventricular communication, or VSD, ventricular septal defect, which I know some of your children were born with. So taking it one step further, as I told you, those cells start out in the neural tube and they have to migrate to get to where they need to go. So this is a close-up of a normal developing heart. So this is a cross-section of the neural tube. And you see this stream here, these red stained cells in this experiment are the neural crest cells on their way migrating to form those final structures in the heart. And when we knock out or get rid of the ETS1 gene, this process is disrupted, so now you do not see that nice stream of cells and they're kind of scattered randomly. So that was really exciting because that helped us to understand now how loss of ETS1 in this situation can cause VSDs. And again, just to summarize, as I mentioned before, there's a very specific group of these neural crest cells that have to migrate all the way down here to form that, help form that wall separating the left and right ventricle. And I don't know, yeah, so just to go back, what we have been able, oops, why am I going the wrong way here? There we go. What we've actually been able to show is if we culture these cells, we can show that they move more slowly, so that's what we refer to as a migration defect, and they're actually stickier. So they basically just get stuck. They can't get anywhere, and that's what you're left with. So this is cool, but remember what I told you in the beginning, my interest, among others, is hypoplastic left heart syndrome. And to this day, I have never seen hypoplastic left heart syndrome in these mice. So why is that? And that brings me to the other half of my talk, looking at the other population of cells that ETS functions in, called the cardiac endothelium. So within the heart, if you think of the heart as a very fancy type of blood vessel, blood vessels are usually made up of cells that make up the walls called endothelial cells. Well, the heart isn't just any old blood vessel, obviously. It's very, very specialized. And it turns out there are 2 subpopulations of endothelial cells in the heart. One is the lining of the heart, the inner wall, the lining of the inner part of the heart that hopefully you see here. And that inner lining is called the endocardium. So the inner lining of cells of the heart. The other population of cells that you can appreciate, let's say down here, is in this outer part of the heart. You see tiny little blood vessels. And those blood vessels are actually the tiniest branches of the coronary arteries. So you actually have ETS1 actually expressed in endocardial cells in this region, and then you have ETS1 also expressed in these coronary vascular cells. And I also want to point out, you can appreciate here that there are sort of 2 compartments of this heart muscle. So this outer compartment that's kind of nice and smooth, a semicircle, that's called the compact zone. And that's actually the part of the heart that does all the contracting. The other part of the heart where the endocardium lines is these finger-like projections, and that's called the trabecular myocardium. In the mature heart, you generally just have a compact zone, and then this trabecular myocardium kind of goes away. And again, just to remind you, we had shown already that ETS1 is expressed in these two populations of endothelial cells in the heart. So the next obvious experiment is to see what happens if we just take out ETS1 in the cardiac endothelium, and even more specifically in just the endocardium or just the coronary vascular endothelial cells. So I want to give you an example of what happens when we take it out in both of those endothelial populations. So starting with a normal mouse heart, I think you can appreciate in this box this kind of compact, dense layer. Again, that's the working part of the heart, the muscle that actually contracts. And I think you can appreciate a little bit sort of these finger-like projections. And so if you look at under a higher magnification, this little box blown up, you can appreciate this area here is pretty dense, so we call that the compact zone, and then you have these other sort of finger-like projections. When we knock out ETS1 just in those two populations of endothelial cells, we see a very, very abnormal heart that in some circumstances, the embryos die even before birth. And compared to the normal heart, we see here almost a complete loss of the compact zone and this very sort of lacy arrangement of kind of the remnants of the trabecular layer. So very, very abnormal. And without that compact zone, you can appreciate why even in the womb, these mice die. There's no ability for the heart to contract. And we can actually characterize those hearts a little bit further. So just as an example, let's see here. So you can see here the normal heart on the left and the mutant heart on the right. And compared to the normal heart, the trabecular layer, TM, is wider, and the compact zone, the CM, is narrower. And just to look at that in further detail, this is now, if you just focus on these two panels, this is a blow-up of what we're seeing there. But this time, green is labeling heart muscle cells. The blue is just the nucleus. And the red here is labeling those coronary vascular endothelial cells, the very, very tiniest vessels. Now when we look at the mutant, if you look carefully, all you have to do is know colors, so hopefully nobody is colorblind in this room. But compared to the wild type, you can appreciate how this compact zone is much narrower, and you can also appreciate where you see these red cells lighting up, you see almost a complete loss of those coronary vascular endothelial cells. So that's very high up, but what we also notice is compared to the controls, these branches are all branches of the coronary arteries, so the arteries that supply blood to the heart itself. And compared to the control, in the ETS1 knockout, you see much less branching and much fewer connections. So what we could do is do a molecular analysis to try to understand what are the genes that ETS1 is regulating. And it turns out, fortunately, when we do something called RNA sequencing to look at all 23,000 genes in these heart cells to see which ones are affected, first of all, just looking sort of qualitatively, we see that consistent with our findings, endothelial development, genes involved in endothelial development, genes involved in blood vessel formation are affected. We also see that genes that encode factors that are in the spaces in between heart cells are also affected. And all I'm going to show you here is that we could actually identify direct targets of this ETS1 transcription factor. And if I go on to the next slide, what I can show you is, I'll just give you this one example. Let's look at this one. So this is one of the direct targets that ETS1 regulates in the developing heart, again, in the endothelial cells and the endocardium. And we know that in some people with Jacobson syndrome, they have a heart defect called tetralogy of Fallot. And in this case, we know that mutations in this one gene have been found in patients with tetralogy of Fallot. And that's a really interesting observation because it tells us for any given patient, for whatever reason, this gene's function seems to be most sensitive to the loss of ETS1, which would explain why this patient has tetralogy of Fallot versus maybe a different patient that has a bicuspid aortic valve. Everybody with me so far? All right. So the other thing that we could deduce is if that compact zone layer is thinner, it's either because there's death of cells or there's lack of growth of cells. And I'm not going to go through this slide other than to tell you that what we found is that that thinning of that compact zone is due to decreased growth, decreased proliferation of the cardiac myocytes. And looking at a mouse heart, we wanted to get even more functional information about how is this happening. So this is a normal mouse heart, and what you see here, this dotted line, is the endothelial cells that make up the coronary arteries, those tiny branches, and normally they migrate all the way down to the bottom of the heart. So that's a normal mouse heart, embryonic heart. And when we knock out ETS1, there's a failure of those cells to make it all the way down, and that actually helps us to understand why we didn't see any of those vascular cells on that previous slide where the red was lost. We could also do studies to test how ETS1 actually affects the development of those blood vessels, because remember I showed you the coronary arteries were very abnormal, even grossly. And compared to a normal control, we found that if we inhibit ETS1 in cultured cells that will normally form these little tubules and little miniature blood vessels, if you knock out ETS1, that process is interrupted. So impaired migration and impaired ability to form blood vessels is what we think likely is the cause of that compact zone being so much thinner. So this work we published back in 2022, and now I'm going to give you an update to actually take this one step further. And so I've told you that, again, there's the endocardium and there's the coronary vascular endothelium. So here's the coronary vascular endothelium, and here's the endocardium, the inner lining of that heart, and these little things here are depicting the coronary vascular structures. And an amazing technology that we took advantage of is something called single-cell RNA sequencing. And compared to a wild-type control, what we could do is take a piece of that heart, and at the individual cell layer, we could assess the expression of all 23,000 genes. And we do this for thousands of cells. And Lou Wang from my lab has shown an incredibly interesting observation that these endothelial cells are actually secreting growth factors. And that would explain why the absence of ETS1 causes a loss of these growth factors and decreased proliferation and growth of that compact zone. All right. That's pretty much what I'm showing here. But taking it one step further, what Lou was able to show is if he took a heart that's missing ETS1, that has that abnormal thinned compact zone, he could actually add back these factors to that heart in a petri dish and show that, so if you take the heart from an ETS1 knockout mouse that would otherwise have that thin compact zone, if you take that tissue and treat it with these growth factors, guess what? It restores the growth. So that kind of completes the circle to show that ETS1 is regulating growth factors that are essential for promoting the normal growth and development of these heart cells. So that's what we've learned in the mouse. But here we are, I've only got about a few minutes left, and I still haven't shown you a mouse heart with hypoplastic left heart syndrome. That disease that I just described for you is something called non-compaction of the ventricle. It's also a defect in how the ventricle develops, but it's not hypoplastic left heart syndrome. But what I want to show you is that sometimes if you don't get answers that you're looking for in a more complex animal model system like mice, we turn to some simpler model systems to see if we can get a better understanding. And in this picture, what I'm showing you is what happens if we specifically eliminate ETS1 in a developing frog heart. So just to orient you, this is a cross-section of the ventricle of a normal frog heart. And you see these kind of finger-like projections, so that's the trabecular layer. It's pretty early on, but the compact zone is this outer layer. And in this case, when we delete or knock out ETS1 in the developing frog heart, finally, 45 minutes into my talk, I can tell you that we have a very abnormal small ventricle. And what you can see here is compared to that very nice organization, it's almost like this ball of cells. And the other thing that's very interesting is, if anything, maybe there's more of this compact zone, but we've lost all those finger-like projections. This is what's been done in frog hearts, and we've also replicated this in zebrafish, which is another simpler animal model that we use for studying genes in heart development. We could actually take it one step further, because if we think the endocardium is one component that's critical for secreting these factors, we could actually do an experiment where we just destroy the endocardium as the heart is developing. And that's what I want to show you here is what we did. Just to focus on, here is a normal mouse heart early on. And if you ablate or get rid of that endocardium compared to that normal mouse heart, these mouse embryos actually die very early, but you can see the ventricle is much, much smaller. And whereas here you see these finger-like projections, you pretty much lose them. So all this is telling us that probably it's some combination of endocardial cells secreting factors and coronary vascular endothelial cells secreting factors that are necessary for the ventricle to develop. And that's just, again, what I show here. So this sort of dual lineage or dual pathway, endocardial cells secreting factors that are ultimately required to promote heart cell proliferation, and similarly, coronary vascular endothelial cells by a different pathway, including these two genes that we just discovered that have never been reported before to promote heart cell proliferation. So before I get to that, oops. These experiments have been done on mouse embryonic hearts. I'm going to go off on a little bit of a tangent for a moment. So as I said, we identified these two novel heart growth factors that are required for the heart to develop normally. So if I start having chest pain right now, and you rush me to the hospital, and they diagnose me with an acute myocardial infarction, the first thing they're going to do is rush me to the cardiac catheterization lab and use a catheter to deliver drugs that will open up my blocked coronary arteries. The problem, though, is once you get to that point, you already have suffered damage to your heart tissue. And if it's bad enough, your heart will fail, because normally when heart tissue is damaged or killed off, it's not replaced with new heart tissue. It's replaced with scar tissue. And obviously a scar cannot do the work that a cardiac myocyte is supposed to do. So this kind of comes back to the point I made before about how studying something rare might have broader application. What we are really hoping to do is not only to understand, and I'll come back to this in a moment, how we think loss of these genes cause hypoplastic left heart syndrome, but by identifying these growth factors, we might be able to change sort of the dogma, and the dogma is adult heart cells cannot regenerate, cannot heal. And maybe with the right combination of these factors, and we're not the only ones doing this, but we discovered these two, that maybe this could be a new therapy to target not only that non-compaction ventricle, but maybe even a much more common disease, namely adult myocardial infarction. Stay tuned. So lastly, what I want to show you is the studies. Close your eyes if anyone doesn't want to see this. Coming back to this baby's hypoplastic left heart syndrome, Jacobson syndrome heart. And really very little is known about what this tissue in that very tiny left ventricle looks like. And we've begun to do some really interesting studies. And compared to a 13-day-old baby that died from some non-cardiac cause, if you see these kind of railroad track structures, that is actually the contractile apparatus that allows the heart to squeeze. And that's called the cardiac sarcomere. And when we took tissue from that Jacobson baby's hypoplastic left heart, from actually either the left or the right ventricle, we have some really interesting observations here. One is that it looks like, as you can imagine, these blue dots are the cell nucleus. There are a lot more blue dots here than here. And it looks like the cells are all sort of compacted together, right? The other thing is, again, so two observations. We don't see those nice sarcomeric structures, and they're kind of compacted together. So we think that's beginning to give us some insights into how hypoplastic left heart syndrome occurs in Jacobson syndrome. And this is just sort of quantifying that. And we see the same thing actually in the right ventricle. And remember what I showed you in the mouse when you didn't see those red signals in that compact zone? This is staining of the tissue from that same heart, looking at coronary vascular endothelium. All these red structures here, like this is a cross section, this is a longitudinal section of of the coronary vessel branch in that heart. These red dots are actually background, they're red blood cells. So just like in the mouse heart, in this case, what we see is a complete absence of the coronary microvasculature. So why is that important? Well, for one thing, you need the coronary vasculature to supply all the nutrients for that heart tissue to develop. But the other thing that I just showed you is that if you have less of those heart cells, you're also going to have less of those endothelial cells, you're also going to have less of the growth factors secreted. And that now becomes a very plausible mechanism to help us understand how ETS1, and probably at the end of the day in combination with FLY1, lead to impaired endocardial function and impaired coronary vascular endothelial function, giving you that very hypoplastic ventricle that I just showed you. And that's what I just pretty much summarized here. What we don't know, though, which we have some guesses, which is one of our future challenges, is why don't we see hypoplastic left heart syndrome in the mouse heart, whereas we do in those simpler animal models? Two things. There seems to be a correlation between the hypoplastic ventricle and whether or not you have that trabecular myocardium, those finger-like projections. So we think, and I don't have time to go into details, that you probably need the trabecular myocardium to develop to help the rest of the ventricle develop. The other component to this is we think that in the simpler organisms, you just need to knock out the one gene, ETS1. In humans and mouse, we know that ETS1 and FLY1 together, well, let me rephrase that. ETS1 and FLY1 have similar functions, but not identical functions. And with that in mind, we think that ETS1 and FLY1 may regulate some unique genes that neither one entirely does alone, such that we think that in the mouse heart and the human heart, you have to lose both ETS1 and FLY1 to get the hypoplastic left heart syndrome rather than the non-compaction. And to strengthen that argument, I mentioned before that we now know of a patient with a mutation in FLY1 that has hypoplastic left heart, so that strengthens that argument. And this is just to show you that actually there are some differences between the structure of ETS1 and FLY1. This AD stands for activation domain. And so we only have one activation domain in ETS1 and two in FLY1. And we think that that might be the critical difference that requires loss of both to give you hypoplastic left heart. So this is pretty much what I've summarized for you already, that we think both genes have a critical role, and if you have overgrowth of the trabecular myocardium, that gives you the non-compaction that we see in the mouse. If you have loss of trabecular myocardium, that gives you the hypoplastic ventricle. All right, so I wanted to add this slide because this is pretty much the whole talk. Just to kind of summarize, about half of everyone with Jacobson syndrome have some kind of a heart defect, including a very high frequency of hypoplastic left heart syndrome, and specifically of this one anatomic subtype. We've identified ETS1 and FLY1, we think, as candidate genes. And what we know is, based on our studies on ETS1, it's expressed in two populations of cells in the heart, namely the neural crest and the cardiac endothelium, specifically endocardium and coronary microvasculature. But interestingly, it's not expressed in the cardiac myocytes. And so we think that there is, at least what we have shown in the mouse, is that there is a dual contribution from both the endocardium and the coronary microvasculature, such that each of those populations of cells secrete growth factors that together are required to form a normal heart. And our most recent studies on the human heart show decreased coronary vasculature, which, as I mentioned, begins to give us a very plausible mechanism for how you get that underdevelopment. And I am hopefully, maybe next time, if I'm lucky to be back here in 2 years, we'll be able to answer for you this question as to why it depends on which species you take out ETS1 to determine which type of problem with the development of the ventricle occurs. We are actually paying a company $23,000 as I speak to generate a mouse that has both ETS1 and FLY1 missing. So stay tuned. And I just want to, again, acknowledge you guys and a number of other foundations that have been just amazingly supportive. And I can tell you absolutely that without your support and these other foundations, I would have nothing to tell you about today. So this is huge. These are the most recent sources of funding. Just to reflect on the fact that this picture, this slide actually shows 6 of some of the most contributory patients, children with heart defects. And actually, sadly, as I mentioned, Edgar passed away October of last year, and all these other patients have also passed away. So some of the hardest situations are also, to us in research, some of the most informative situations, and I am truly forever grateful to all these children's families who have been so amazingly supportive, particularly in their most difficult times, to want to help create a legacy for their child. And this is why, for me, it's really important to show this slide, because obviously we wouldn't be where we are without these children. And finally, I want to acknowledge some incredible people that I work along with. So the superstar in my lab, this is Lu Wang. So Lu Wang is a PhD scientist who joined my lab about 8 years ago from China. His background was in vascular endothelial cell biology. And when he joined the lab, I was really thinking it must be due to defects in the neural crest cells. Well, it's his wisdom and perseverance that's shown me that at the end of the day, it's probably that cardiac endothelium that's the real smoking gun in this process. Lou is absolutely amazing. And his last paper just came out, the most recent work that I showed you, just 2 months ago. So kudos to him. Basically, almost all the work that I showed you today has been done by Lou. Shui Ni is actually a PhD researcher, colleague of mine at Georgia Tech, who has done all the work in the frog heart. And actually, many years ago, she was the one that showed that it's probably the endothelial lineage in the frog heart that's affected that gives that hypoplastic ventricle that I showed you. And finally, I want to acknowledge Rolf Bodmer and his wife, Karen Oakord, 2 brilliant PhD scientists studying heart development. They actually work on fruit fly heart development and zebrafish heart development, and their contributions have been amazing to help understanding how, by using those very simpler organisms, we're able to now identify what we think are some of the problems that we can then ask in the human heart and in the mouse. And again, a tribute to Edgert. I hope, like I mentioned the other day, that we can start a memorial fund, some kind of a scholarship fund at his high school to support new college students that are interested in doing something that helps children with special needs. And I think that's it. Thank you. Is there a question from? There's no mic.
No, they didn't hear what I said. So it's basically meeting outside. I just said let's meet all in 15 minutes outside for the group picture.